Woman Jekyll, welcome everyone to today's Wednesday seminar where we have Dr. Andre Samson from my lab and Edwin Hawkins' lab presenting. Firstly, I'd like to acknowledge the traditional owners and custodians of the lands on which we meet today. So for me and those in Parkville and many of us, it's the lands of the Wurundjeri people of the Kulin Nation, but we also pay our respects to elders of other communities elsewhere, wherever you may be. And we acknowledge their continuing custodianship of the land for many, many years. So it's a great pleasure to introduce Andre. So he's been working with Edwin and I for more than 3 years now. He joined us from Sean Jackson's lab in Sydney. And before that, he'd trained at Monash with Rob Medcalf and also in part with Steve Bottomley. So for those of you who know those individuals, you would realise the diversity of skills that he brought to the table. So everything from understanding of protein aggregation in chemistry through to microscopy, neuronal science, mouse behavioural science, and many of you have benefited from his knowledge in these areas, including the work on neurons, tissue clearing. And what you're going to see today is really, I think, it's a sort of an accumulation of these skills that allows him through a chemical knowledge to sort of make these key insights into really important processes. And as you'll see from this very careful work, how powerful that knowledge is. So without further ado, Andre, fire up.
Thanks very much, James. Can you hear me? Yep. Great, wonderful. All right, so I'll jump straight into the seminar. So as I'm sure you're all aware, eukaryotic cells have an array of genetically encoded programs that finely and strictly control cell death. Apoptosis, of course, being the most recognised and best understood form of programmed cell death. And collectively, these programs of cell death are essential for the development and survival of multicellular organisms. And as we heard from Marcel a few weeks ago in the Wednesday seminar, there's a growing appreciation for the mechanistic crosstalk and functional redundancy and flexibility that exists between these various programs, so that they operate as a networked unit that can really achieve cell death whenever and wherever it's needed in an altruistic sense for the better good of the organism. So today's seminar, I'm going to focus on one of these programs of cell death called necroptosis. So necroptosis is a developmentally dispensable form of programmed necrosis or necrotic cell death. So that's a key point of difference with apoptosis. And also, unlike apoptosis, necroptosis is caspase independent. And whereas in apoptosis, you've got largely a preserved integrity of the plasma membrane to allow the intracellular constituents to be catabolized, necroptosis is quite different. It results in frank and overt lysis of the plasma membrane and release of intracellular constituents into the extracellular space. And so because of this, necroptosis is thought to be largely immunoreactive and spark autoreactivity, and also be pro-inflammatory via the release of DAMPs, or damage-associated molecular patterns. So necroptosis is first thought to have come about ancestrally as a defensive measure against infectious agents that can block apoptosis. So basically it's a fallback measure of sorts. And consistent with this idea, many virus and bacteria can either positively or negatively regulate necroptosis. In fact, some of them have encode for proteins that directly commandeer or subvert the necroptotic pathway. And here's just a short list of some of the diseases or agents where necroptosis is thought to be a key. But beyond communicable diseases, necroptosis is now appreciated to also play a role in many non-communicable diseases. And here's a summary of some of the diseases, and by no means all of them. And so you can see necroptosis is thought to play a significant role in human health and disease. Of direct relevance to today's talk is the role that necroptosis is thought to play in inflammatory bowel disease. The work that we do has direct implications for that. And I think the clearest example of the role that necroptosis plays in inflammatory bowel disease is the fact that mutations in the necrotic pathway are monogenic causes of very early onset inflammatory bowel disease. So I think that's clear that that's an indication of great interest. So the term apoptosis was actually coined back in the early 70s by an Australian pathologist by the name of John Kerr. And in the intervening 50 years or so, many anti-apoptotic or pro-apoptotic drugs have made it into clinical practice, and venetoclax is the one that's closest to home. By comparison, the term necroptosis was only coined in 2015, and many of the key understandings of the pathway have come since then. So owing to this recency and the promise of this new cell death modality that hasn't been targeted, many pharmaceutical companies have embarked upon discovery programs to develop anti or pro-necroptotic agents. And so we're talking GSK, the Chinese Ministry for Science and Technology, Denali, Sanofi, AstraZeneca, and indeed many of us here at WeHi together with Naxos Pharma are looking at bringing the first necroptotic targeted drug to market. There are a variety of inflammatory stimuli that can trigger necroptosis, and here's a broad summary of necroptotic signaling. So you have anything from the TLR stimuli to the death receptor ligands, interferon, or nucleic acid-based danger signals. These are all being shown to elicit necroptotic cell death via their actions on their respective receptors. Now, there's a lot of crosstalk between these receptors, so they tend to quite often work in concert with various stimuli that all then converge onto this core signaling pathway. So this is the key part here. The core necroptotic signaling axis, as I'll call it, involves RIPK1, RIPK3, and MLKL. RIPK1 and RIPK3 are canonical kinases. A variety of these stimuli leads to the autophosphorylation of RIP1, which then phosphorylates RIP3. RIP3 is also, like I said, a kinase that then phosphorylates MLKL. Now, MLKL is a pseudokinase, so it doesn't have phosphotransferase activity, but its phosphorylation is sort of a flagfall event in the pathway. And this phosphorylated MLKL is really the terminal effector of the pathway that then directly goes on to perturb cell membranes and cause cell lysis. The other point that I want to make is that RIPK3 and MLKL are the only obligate components of necroptosis. Every other part to the pathway in some way, shape or form can be dispensed of. And the other point that I want to make is that there's a surrounding network of modulators that finely tune the actions of the receptors and the flux of signaling along this core axis. And this is a thesis that a PhD student in the lab, Sarah Garnish and I are trying to put forward in a review that we've submitted. And it's something a theme that will come up at the very end of the talk, is that, and there's this largely context-dependent network of proteins that involve E3 ligases, dubs, phosphatases and kinases, catabolic machinery, endosomal and exosomal machinery that all basically modulate the signaling activities of this core pathway. So in essence, it's actually a very simple cascade that has a very complex network of tuning processes. The canonical form of necroptosis, so the form of necroptosis that is most widely studied and best understood is TNF-induced necroptosis, and that's the type of necroptosis that we primarily study in the lab. So an abridged version of TNF-induced necroptotic signaling is shown here, and I've back-ended this, so I've looked very much at the terminal stages. We're most interested in MLKL, and I apologise to those that are more interested in the upstream events. I've summarised in the entirety of the upstream signaling in this one arrow here. But basically, TNF-induced activation of the TNFR1 receptor under conditions where the cellular inhibitors of apoptosis proteins, the CAAPs, and caspase 8 activities are suppressed or reduced. Activation of this pathway leads to the formation of this signaling platform called the necrosome. And it's called the necrosome is a amyloid-like assembly of RIPK1 and RIPK3, and this core recruits MLKL to it, where RIPK3 can then phosphorylate MLKL. Once phosphorylated, this causes a conformational switch in MLKL, which liberates the N-terminal domain, promotes the formation of phosphorylated oligomers of MLKL, and these phosphorylated oligomers of MLKL then have the propensity to associate with membranes and disrupt their integrity, thus causing membrane lysis and cell death. To induce this pathway in the lab, we use a cocktail of drugs that I'll refer to today, or a necroptotic stimuli that I'll refer to today as TSI, and that's TNF, smacomimetic to reduce the levels or activities of the IAP family, and the caspase inhibitor enricocin, which I'll refer to as I, so TSI. And when you do that, you get canonical activation of this pathway. Now, I should say that essentially all of the information that I've just described to you has been fleshed out using biochemical assays and fractionation assays, say Western blots, immunoprecipitation, mass spectrometry, together with lots of genetics. But there's still a lot that we don't know about this pathway. And here's some of the leading questions in the field. How is MLKL recruited to the necrosome? Where exactly in the cell is the necrosome? And once MLKL is in its fully activated state, what does it look like? What is the toxic species exactly? These oligomers, are they tetramers? Are they trimers, octamers? A variety of different, or even amyloid species have been reported. How does MLKL eventually make its way to membranes? How does it translocate to membranes? And when it is on the membrane, how does it disrupt these membranes? So there's still a lot of really critical pieces of the puzzle that we're yet to put together. As I said, most of the elucidation of that canonical pathway is based around bulk fractionation assays or mass spectrometry or the whatnot. But really, what hasn't been done is we wanted to take a different approach at looking at how this pathway is activated. And to that end, we've been putting together a toolbox of antibodies to enable the imaging of endogenous necroptotic signaling in both mouse and human cells. So this is something we've been doing this year, and I'm pretty happy to be presenting it. I was hoping to have the manuscript that involved this toolbox, or outlining all of these antibodies that we have to the basal and phosphoforms of RIPK1, RIPK3, and MLKL for both mouse and human cells. I was hoping to have this on bioarchives in time for this talk, but you'll have to wait another week. So for those that are interested, you'll have to look at bioarchives next week probably. But so like I said, we've got antibodies that now can be used to image via immunofluorescence the basal and phosphoforms of RIPK1, RIPK3, and MLKL in human and mouse cells. And really, to complete this toolbox, we've had to develop a range of monoclonal antibodies to a number of the mediators, and they are shown here in the asterisks. So these are WeHi-based in-house monoclonal antibodies that we've had to develop to complete the toolbox. And just a technical point from the paper that we're putting together right now is that, so compared to a commonly used commercial antibody for MLKL, there's a real need for better antibodies. As you can see from an imaging perspective, this commercial antibody, although it works for Western blot, it's totally non-specific from an imaging immunofluorescence perspective. And the other technical point that's really driven the selection of good monoclonal antibodies is the understanding that we cannot use cross-linking based fixatives. And the reason for that, we believe, is that MLKL's got this incredibly high lysine content. It's got a lot of functional lysines. And when you fix MLKL with, say, paraformaldehyde or formalin, you basically remove its immunoreactivity, and it's very hard to salvage. So using non-cross-linking fixatives is one of the key steps that's allowed us to basically screen and identify a whole bunch of monoclonals for the necroptotic pathway. So now with this toolbox, we've been using it to get a new look at how necroptosis unfolds in cells at an endogenous level. And so for the rest of the talk, I'm going to focus primarily on work that was published in June earlier this year. We're using this toolbox. We've tried to address two questions in the field, which was, how does MLKL move from necrosomes to the plasma membrane? And what happens when MLKL does reach the plasma membrane? So these are questions that we've been managed to address using microscopy. So microscopy, in a sense, has sort of come to the rescue to address these questions. So those two terminal steps, MLKL moving to the plasma membrane and what happens, they're very important terminal steps that are really black boxes. And this data set here really highlights how important those terminal steps are and how technically we haven't been able to address them until recently. So this is a simple experiment using our workhorse cell line, our HT29 cells that are widely used by us and the field in general, and we expose them to a necroptotic stimulus, and we basically do a Western blot for phospho-RIP3 and phospho-MLKL, looking at pathway activation. And you can see MLKL gets robustly activated very early on, 4.5 hours after the exposure of a stimulus, and this is plotted here or quantified here in blue. But cells do not begin to die for many hours. So there's this temporal gap from when MLKL is activated at a biochemical level to when cells die, and so this is really the gold standard used by the field, or the gold standard, but one of the most commonly used markers of pathway activation. And you can see it precedes, but by no means accurately tracks the onset of necroptotic death. Even when we use our best assay available to us, which is Blue Native Page, which picks up the movement of MLKL from this lower molecular weight, non-phosphorylated species in the cytoplasmic fraction. When you induce necroptosis, it then translocates to this phosphorylated species up here at a much higher weight, so it's an oligomer in the membrane fraction, and that's our best marker for pathway activation. You can see even when we do that, from a biochemical perspective, MLKL is fully acted hours before cell death even begins. So this gap between MLKL activation and cell death is not a particular curiosity of the model that we've chosen. It's a common feature reported, as you showed here, with different stimuli and different cells, but has been reported by others in the field. So it's a common feature of necroptosis. So the point here that I want to just dwell upon is that necroptotic cell death occurs hours after MLKL becomes fully activated by the necrosome. So this really suggests that there's some very important rate-limiting checkpoints that occur downstream of its biochemical activation, and that current methods provide really little insight into these terminal steps. And that's exactly where an immunofluorescence microscopy approach has proved to be a bit of a game changer. So this was the chronology of the pathway circa last year, and we've been able to extensively add to our understanding of that. So I'll step you through how this looks like at the microscopy level. So we have, these are two separate monoclonal antibodies raised against MLKL. One's commercial and one's in-house, and they pick up different states of MLKL, and this will become clear as you go on. So under resting conditions, MLKL resides, is diffusely spread throughout the cytosol, and if you use super resolution microscopy, it's actually existing as very small punctate structures all the way through the cytosol. You then stimulate the pathway with TSI in our case, but you get necroptotic stimulus, and what actually happens is MLKL disappears from our radar. What do I mean by that is I don't think we have an affinity reagent. We don't have a monoclonal antibody that's able to pick up these stages in the pathway. So a simple explanation for this is that in these stages, RIPK3 is probably bound to MLKL. RIPK3 engages this part of the MLKL molecule. This is the C-terminal domain of MLKL, and where RIPK binds MLKL overlaps with where our monoclonal antibodies recognise MLKL. So we think there's basically the binding of RIP3 to MLKL sterically prevents our ability to pick up these steps in the pathway. So this is when MLKL is at the necrosome being phosphorylated and oligomerized. And the data that really demonstrates this clearly, it was published by Emma Petrie, a postdoc formerly in the lab in PNAS earlier this year. And this was work done in collaboration with the Sharidi lab at NYU. And together we've generated a library of approximately 40 monobodies. Monobodies are antibody-like or synthetic ligands that are antibody-like in nature. They're built around the fibronectin scaffold, and they've been selected for their ability, in our case, to bind to selectively to human MLKL. So the monobody that they can be expressed by the cells, and then when they're expressed by the cells, in our case, they'll specifically bind to MLKL. Now the monobody that I'm talking about here today is monobody 37. It has a reasonably high affinity for the N-terminus of MLKL, and it does a specific blockade of the pathway. It doesn't, when you express monobody 37 in cells and you stimulate with necrototic stimuli, you get robust phosphorylation of MLKL. So MLKL is recruited to the necrosome just fine when monobody 37 is expressed. It's phosphorylated, it oligomerizes just fine, and here's a Blue Native page showing that. So this is endogenous MLKL oligomerizing and shifting to the membrane fraction. In the presence of monobody 37, as shown here, MLKL also oligomerizes just fine when it's stimulated, and the pathway is stimulated. But in the presence of monobody, you can see a monobody 37, you can see MLKL is not translocating to the membrane fraction. It's getting stuck in this cytosolic fraction. So we've got a specific blockade in the pathway that prevents, when monobody 37 is expressed, MLKL doesn't associate with membranes. So if we go back to our microscope assay and we stimulate the pathway in the presence of monobody 37 and we look at this by immunofluorescence, really MLKL looks discernibly no different to as if we hadn't treated the cells with necrotic stimuli in the first place. So this is why we think we're missing an important piece of the chronology from an immunofluorescence perspective. We know that these cells have bucket loads of phosphorylated oligomeric material. We simply can't pick it up. If, in the absence of monobody 37, you allow MLKL then to transition to the membrane fraction, what do we see? We see two hallmark structures of MLKL activation, and I'll zoom in on one of these. So this is a fairly low-res image of these cells, and what we're looking at here for this one antibody is picking up these cytosolic clusters of MLKL, as I refer to them. So these are where MLKL is phosphorylated, oligomerized, and membrane associated. There's another state, these are called hotspots, picked up by another antibody against the phosphosite in MLKL, and we'll refer to these as hotspots. This is a saturated hotspots, but it also exists as some of these small structures internally. So both of these flavors of MLKL are biochemically fully activated. One's internally in a perinuclear space, and one is going to sit on the periphery of the cell. So I'll explain that in a bit more detail. The kinetics of the clusters and the hotspots of MLKL that form, they nicely mirror the lysis of the plasma membrane. As you can see the formation of these two structures really gets going when cells are entering that terminal lytic phase of necroptosis. And they increase not only in frequency, but also intensity and also size. So they seem to be growing and stimulating as the cells are reaching that threshold for death. So we think we've now got these nice microscope-based readouts for these events downstream of biochemical MLKL activation. So we've got readouts that are microscope-based for the terminal steps in the pathway. So a little bit more about clusters. We think these clusters represent necrosomes, and there's a variety of reasons for that, but the main reason is that they preferentially co-stain for very small deposits of RIPK1. RIPK1 being, of course, the necrosomal initiator of the necrosome. So I hope you can appreciate here these magenta clusters here, and the color coding will stay the same throughout the talk. These magenta clusters here are co-stained for the presence of RIPK1 in cyan. And here's a zoomed in example of one of these clusters. And then emerging from these clusters appears to be the phosphoforms of MLKL, and I'll explain that in the next slide. So those cytosolic clusters, if you zoom in on one of them here, you can see from there you get an emanation of what's that terminal step. This is another antibody against MLKL. So this is now from these cytosolic clusters that co-stain for RIP1, seems to emanate these small vesicle-like structures of what is the terminal steps in MLKL. So it's recognised now by this late antibody, this phospho antibody. So MLKL seems to depart from these clusters and these small vesicle-like structures, which then accumulate as these quite striking hotspots. And here you can say these hotspot here co-localizes very nicely with the wheat germ gluten stained plasma membrane. So there seems to be this directionality to the clusters and the hotspots, and that will become more apparent in this video here. So this is now a 3D movie showing the clusters and the hotspots and the directional relationship between the two. I'll just play the video, get rid of the membrane. Now we're going to zoom in on the clusters and the hotspots, and you should be able to see that the clusters are closely abut the hotspot. And the other thing that's quite striking is the hotspots have a very unusual or atypical appearance to them. So James likens it to a cut lotus root, and I think that's quite apt. Sometimes they're rings, sometimes there's this cut lotus spongy-like appearance, but they're very large, quite irregular shaped structures. So now we've got this revised chronology of necroptosis, where upon activation, you get this functional unit, this necrosomal cluster that forms where MLKL is phosphorylated, oligomerized, and where it associates with membranes. And this is on an internal membrane. That's an important point of difference. MLKL then emerges from this necrosomal cluster in its fully biochemically activated state, and it's shipped to the periphery in what appears to be vesicles. And once it gets to the vesicles, it undergoes another compartmentalization event to form these large supramolecular micron scale hotspots, where we believe it disrupts the membranes. So this really threw up three obvious questions that we were really interested in tackling. So first of all, most importantly, are these hotspots where MLKL is doing all the damage? Is this where MLKL is killing the cell? Why on earth does MLKL form hotspots? It hadn't been described before. That's a new structure. And how is MLKL relocating from these internal compartment to the periphery? How's it getting to the plasma membrane in the first place? So the first point. And this is work that Ying Zhang and Noel Geoghegan tackled using lattice and light sheet microscopy at first. So this is a separate experiment where they looked at membrane damage using a Nexin. So a Nexin 5 binds to the externalized phosphatidylserine as a surrogate marker for membrane damage. So you'll see an Nexin in yellow. You'll see the uninjured membrane is pre-stayed in the magenta colour, and the moments of cell death pop up in cyan when the cell becomes permeable. So you can see straight away that the first thing that forms are these hotspot-like annexin structures. So annexin is forming hotspots, and then this hotspot persists, and then you get ongoing perturbation of the membrane. And you can see the membrane here is quite deformed in and around that hotspot. You end up with these quite striking bubbles that seem to be cell resident. So I don't expect you to take that all in. I'll move to stills in the next slide. And so you can see, and I should say this is the most fun data set and the richest data set I've ever been handed. I spent 2 weeks with a bucket of popcorn going through all of these lattice light sheet videos, basically just categorizing and analyzing how cells die. So it was an amazing data set that Ying and Noel were able to generate. So you can see here, you get this initial emergence of a hotspot, and that hotspot persists. You get local perturbation, so it seems to be an area where there's a lot of membrane dynamics. Eventually, there's this moment where the cell can not withstand that damage anymore, and you get this sort of overt or cataclysmic rupture of the plasma membrane. And these frames here are 2 minutes apart, and you can see a zoom in here. So proximal on the leading edge of those hotspots, you get this major breach of the membrane, and this is a common theme. So looking at cells, over 80% of the cells undergoing necroptosis present with these Nexin positive hotspots before dying, and that blowout, so this large pop of the membrane in essence, seems to be a common feature. Pore-like small pinpricks in the membrane are much less common, and then there's some other nondescript ways that the cells seemingly die, which are quite interesting and worth following up. Okay, so we had this interesting finding where we've got these hotspots of MLKL that are forming in fixed cells. We've got annexin hotspots that are forming in live cells, trying to bring the two using correlative immunofluorescence approaches. So here what we've done is we tracked the annexin, the binding of annexin to cells over time that are undergoing necrotosis, and just before the cells die, we fixed them and then immunostained them and then refound them. And so what you can see here is that there's really good proximity between the hotspots for MLKL that are forming and where annexin initially accumulated on this membrane. And that should be more apparent in this 3D video here, where you can see that there's seems to be a real close physical proximity between the annexin hotspots and the MLKL hotspots. So for us, this was really the smoking gun, and told us that hotspots are the main site where MLKL is damaging the plasma membrane. Okay, so that was one question. Next question is, how is MLKL relocating from the internal clusters to the peripheral, the cell periphery? So to get at this question, we screened a variety of trafficking inhibitors for their ability to block this terminal effector phase of necrotosis. And so what I mean by that is we tried 3 different conditions. One where you add a trafficking inhibitor upfront together with a necroptotic stimulus, or you're adding these trafficking inhibitors very late, so 5 or 6 hours after we know MLKL is already fully biochemically activated. So MLKL is already, when we add these drugs at these points, we know MLKL is already phosphorylated, oligomeric, and on an internal membrane. So to cut to the chase, when you inhibit or destabilize trafficking along the Golgi pathway, along the microtubule pathway, or along the actin pathway, you get this inhibitory profile that extends right through all of these 3 time epochs. So these are 3, and then if you do a cocktail inhibition of all three trafficking pathways, you get quite a profound level of inhibition that at least in the short term, is as effective as direct MLKL inhibition itself. So digging a little deeper, what does interfering with these trafficking pathways do? It doesn't affect the recruitment of MLKL to the necrosome, and we know that because MLKL is phosphorylated just fine when you add these trafficking inhibitors. So you inhibit the Golgi, the microtubule, and actin pathways. When you destabilize those pathways, MLKL is phosphorylated just fine, so it's recruited just fine. And this is an IncuCyte data set across 3 cell lines, which really shows that it doesn't affect its recruitment, but it does instantaneously slow down the rate of necroptosis. So what we've done here is we've stimulated necroptosis in these 3 cell lines, waited for necroptosis to just begin, and then what we've done is we've either directly inhibited MLKL with this compound called NSA, or we've inhibited these trafficking pathways. And you can see we can end up in the short term with a very comparable flattening of the necroptotic death curve. So for us, that was quite clear evidence that what we were doing is we were slowing down the ability of MLKL to get to the plasma membrane. And in support of this, if you do our immunofluorescence approach, you can see that essentially by inhibiting these trafficking pathways, you've depolarized or you've randomized where the phosphorylated MLKL is going to end up. So it still phosphorylates just fine, but it's no longer accruing as these very focal hotspots. It's sort of being distributed more uniformly throughout the cell, so it's not able to grow. And numerically, it reduces the amount of MLKL that leaves as clusters and as hotspots. So these trafficking pathways are selectively used in necroptosis to affect the membrane, as selective as inhibiting Golgi, actin, and microtubules can be. And so this is the data that speaks to that. Here you're looking at now on an IncuCyte, you're looking at the annexin binding, which happens earlier, and cell death in PI. And if you inhibit necroptosis, as I showed before, you slow the binding of the further binding of annexin and the onset of cell death in necroptosis. You do not see the same trends with extrinsic apoptosis, which is probably a closest cousin to necroptosis. So it's a trafficking pathway that seems to be selectively used for necroptosis. And if you immunostain, we profiled where these clusters and these hotspots are residing within the cell, and they do preferentially locate to the actin and microtubule networks. Despite our best efforts, we haven't been able to pin down any association between these clusters and these hotspots to the Golgi, but that's still work in progress. But as you can see here along this microtubule filament, there's these clusters and what appears to be these vesicles of the terminal yellow phosphorylated forms of MLKL en route to this hotspot here. So we've got, in addition to the revised model, not only are we getting this necrosomal cluster where MLKL is fully biochemically activated, but then it's being trafficked actively in a regulated fashion using microtubule, actin, and Golgi-dependent networks to get to the cell periphery. And so the next question that we wanted to tackle was, why is MLKL forming hotspots in the first place? So you would have noticed that the hotspots are preferentially locating to points of intercellular junctioning. And also there's been quite a bit of literature about MLKL going to various other organelles in the cell. So we profiled for, I think, about 22 different structures within the cell, and we ruled out mitochondria, endosomes, proteasomes, lysosomes, desmosomes, adherence junctions, and gap junctions. So MLKL's terminal steps didn't co-localize with any of these subcellular structures. What it did, and was quite apparent, is that it co-localized to an extensive degree with markers of tight junctions. So here I'm using one of them, Z01, but it also co-localizes with occludin and JAM-A, which are 2 other tight junction markers. And the thing to note is that it doesn't co-localize sort of with this pre-terminal step that's recognized by one of our monoclonal antibodies. It's recognized, the overlap exists for this terminal step in MLKL, so this is the phosphosite antibody. And it picks up not only the hotspots here, but also these vesicles that are what we believe are en route to the hotspot. So tight junction proteins seem to be sharing the same same trafficking pathway to get to the hotspots in the first place. So that was our guess at the time, and given the level of co-localization, we thought there might be some functional crosstalk between the two. So just a brief explanation: tight junctions and the zonulin pathway, because they were new to me at the time. So tight junctions are really, they basically staple adjacent plasma membranes together. And they form a selectively permeable structure that regulates diffusion between cells. And this selectively permeable barrier is most recognized for its role in the mucosal barrier, although it's equally important to form vascular barriers. And one of the ways that this tight junction is regulated is via the zonulin pathway. And so this is the pathway here that I'm describing here. One of the key drivers of zonulin signaling that directly acts upon the tight junction is TNF. TNF causes endogenous zonulin release, which stimulates zonulin receptors, which the zonulin receptors then reorganize the tight junctions and destabilize the tight junctions. So they basically lose productive anchorage to the cytoskeleton. And this, of course, then leads to leaky gut, which drives further inflammation and stress, and you end up with this negative cycle of signaling. This is relevant to celiac disease. So gliadin stimulates zonulin release and feeds into this pathway, but also the cholera toxin. Cholera drives this pathway, and that's been shown. And there's two cell impermeable reagents that can antagonize or agonize this pathway, and we use them to work out whether tight junctions had a functional interplay with necroptosis. AT1001, also called larazotide acetate, blocks this receptor, this signaling pathway, and larazotide acetate is actually undergoing phase 3 clinical trials for celiac disease. It's been fast-tracked by the FDA and seems likely for approval probably towards the end of next year. And to stimulate this pathway, there's this cholera toxin-derived reagent called AT1002. So we're going to use that. We use these two antagonists and agonists of this pathway to work out whether there was interplay with necroptosis. And it turns out that there was. So you can see here stabilizing tight junctions with AT1001 significantly slowed down the rate of necroptosis, whereas destabilizing the organisation of tight junctions significantly accelerated necroptosis in our cell models. These quite profound slowing or acceleration of necroptosis had nothing to do with recruitment of MLKL to the necrosome. You can see in the presence of these two tight junction modulators, MLKL is phosphorylated just fine. And in fact, it doesn't affect the ability of MLKL to get to the plasma membrane. You can see here, MLKL is getting to the plasma membrane just fine and forming hotspots under all three conditions. So it seems to be that the structure or the stability or the rigidity of the tight junctions or their organisation is really a rheostat for how MLKL acts at the hotspot. And so I'll explain that in a bit more detail. So what we think is that this hotspot here, it's a rheostat, whereas its organisation controls the threshold at which MLKL can breach the plasma membrane. So if you destabilise this hotspot, MLKL can reach the lytic threshold easier. So you need less MLKL growing into this hotspot to cause membrane lysis. Conversely, if you fortify this junction using AT1001, you need more MLKL growing at the plasma membrane to disrupt this plasma membrane. So we think that there's this rheostat, this lytic threshold that's being modulated. So in short, we've got two new checkpoints that we've added downstream of the necrosome. One's a trafficking checkpoint and one is a threshold checkpoint. So the other question that immediately popped into mind when we saw these hotspots that were preferentially situated in junctions is there a cell extrinsic consequence for accumulating MLKL, this toxic MLKL species at junctions? So everything I talked about before now was in the cell intrinsic role. For these checkpoints, is there a cell extrinsic role? And it turns out that there is. So hopefully this video will do the explaining for me. Cell membrane damage in green is shown via Nexon binding, and hopefully you can appreciate that membrane damage can essentially wick from one necroptotic cell to another. And then you get the later onset of death as the cells become cytox positive in green. And so to get some numbers to this, we actually had to analyse many cells. And so what we're looking at now in this graph here is the spatial and temporal coincidence between death moments. And what you're looking at here is cells that are 1 cell width apart versus 2 cell widths apart versus 3 cell widths apart. So these are direct neighbours and these are non-direct neighbours. And so when you look at the spatial and temporal coincidence between these two groups, only the cells that are direct neighbours seem to have a degree of synchrony between their spatial and temporal death moments. And so to put it into plain English, cells that are direct neighbours undergo necroptosis about 25% quicker than, or in a more coincident fashion than non-neighbours. And this seems to be selectively for wild-type necroptotic cells. If you induce extrinsic apoptosis in these cells, there is no synchrony between the cell death of direct neighbours. And if you look at cells that don't have junctions, so for example U937 cells, which are non-adherent, even if they're direct neighbours, there doesn't seem to be any a coordination between when those two direct neighbours will die. So basically necroptosis has this contagious-like property. So in conclusion, we've introduced a new toolbox that we hope will enable the imaging and analysis of endogenous necroptotic signaling in both mouse and human cells. We've uncovered two new checkpoints. One is responsible for actively transporting MLKL in its fully biochemically activated state from the necrosome to these hotspots. And the second one is a rheostat that controls the lytic threshold, so how quickly MLKL at the plasma membrane can cause cell death. And the other point that's worth mentioning is that these are not universally applied checkpoints. As I mentioned in my initial talk, there's a network that flexibly controls necroptosis. Most of these cells use these two checkpoints, but not all cells that we've studied use these checkpoints. And the other thing that this buildup of MLKL junction seems to give necroptosis a contagious-like property, so it can propagate from one cell to another, and that's consistent with tight junctions being a transcellular structure. So it's not surprising that it's going to have an impact on the other cell. So where are we hoping to take this next? So we're hoping to move this to more disease-relevant models. Together with Marie Foe and Anthony Burgess, we're looking at patient-derived human organoids from the gut. And here's just some initial data that we've got that speak to that. These organoids are sensitive to TNF-induced necroptosis, and you can see here, 10 hours after exposure to a necroptotic stimuli, you've got really good cell death happening in these organoids, and if you stay in for tight junctions, the tight junction network is disassembled in these organoids. This new data that we analysed last night, looking at mouse cells. Obviously, the mouse models of necroptosis are very widely studied and are of great interest. And so we're looking at, with our new toolbox, looking at whether the same findings can be observed in mouse cells. And quite excitedly, we see much smaller but still focal accumulations of MLKL at junctions in mouse dermal fibroblasts. And this is thanks to Joel and Edwin. Some fantastic imaging looking at endogenous MLKL activation in those cells. And some exciting unpublished data from Catherine Davies, a postdoc in the lab, looking at whether this, addressing really why MLKL is forming these quite, unique structures at plasma membranes. She's been using model membranes to see how MLKL binds and interacts with those model membranes. And you can see MLKL is not uniformly distributing to those membranes, but once again forming quite defined, unique focal or ring-like structures. So that leads me to thank the many people that have contributed to this work. First and foremost, I'd like to thank the incredible support that I received from James Murphy and Edwin Hawkins. They've been incredibly generous and given me a lot of scope to build this new project and take things in a new direction that initially was unexpected, but notable, and I hope I've mentioned other people along the way. Catherine Davies, Joe and Nish from John's lab have been fonts of knowledge and have really skilled me up in all things necroptosis. Guillaume Ying, together with Noel, for the fantastic lattice light sheet dataset. In fact, the whole team at Centre for Dynamic Imaging, this is a very imaging focused project, so their skills and expertise are vital. We hire Antibody facility for making some wonderful antibodies. The DivCos for keeping this machine well oiled at WeHi, and of course our funding partners. Thank you very much.
Thanks, Andre. Fantastic tour de force. We've got a few questions, as you might well imagine, that have rolled in. So we'll start with Andreas. So he asked a question about the accumulation at cell-to-cell contacts. He preempted your later data, but but he wanted to know if you cultured cells at low density to minimize contact, if there would be less necroptosis.
So we definitely considered doing that, and we did initially start doing that. They were very difficult to interpret. So I should also say that hotspots form even if a cell doesn't have a direct neighbour. And this is consistent with Catherine's data that we think maybe the hotspot formation is an intrinsic property of MLKL itself. So rather than uniformly going to the plasma membrane, it's going to naturally concentrate in focal spots. But yeah, so we did initially do low density cultures, but it's very hard to there's many reasons why we think that might be a confounding way of looking at it. So I think that the more relevant comparison was cells that don't form contacts versus cells that do form contacts.
Very good. There's a related question from MLKL, which I'll follow up with, which is whether you think that the MLKL from one cell is able to transfer to the neighbouring cell given the time to death. Could you show that with a mixed labeling experiment?
Yeah, so we've actually done that, and that experiment is in our paper. We did co-cultures of wild type cells with MLKL knockout cells and wild type cells with RIP3 knockout cells. And so while there's a few low number examples where you could see small amounts of annexin being transferred to the knockout cell, the upshot is less than 1% of the knockout cells died when they were co-cultured with wild type cells. So I was really excited about the possibility that we could get damage transferring from a necroptotic wild type cell to a knockout cell and kill it, but that doesn't seem to be the case. So it seems to be that both cells need to be primed and undergoing necroptosis for these propagation. And I suppose in a sense that's consistent with the idea that the tight junctions are transcellular structures. So you need both of the neighbours to be participating, I suppose, in that necroptotic handshake in a sense. But it would have been great if it did lead to the death of knockout cells, but that wasn't the case, unfortunately.
Cool. Moving on, Misty's asked, in relation to trafficking of MLKL and its proximity to microtubules, is there any evidence of MLKL binding to kinesin or dynein motor proteins to facilitate MLKL trafficking around the cell?
So there's nothing in the literature on that. I do have inhibitors of kinesin and dynein in the freezer. I just haven't done those experiments, but that is definitely something that's on the to-do list. I would predict so. But I would say that actin is probably, the actin trafficking pathway really does represent the lion's share of the trafficking from the necrosome to the hotspot. So if you looked at the trafficking inhibitor, inhibiting actin-based trafficking with cytokalasin was a really potent way of slowing down necroptosis. So yes, tubulin inhibition does do something, Golgi inhibition does do something, but actin inhibition or actin trafficking inhibitions, we think where it's at mainly.
Very good. A question from Anonymous. Do MLKL binding lipids like PIPs accumulate at tight junctions? I guess more broadly, could you speculate on what you think is happening at the membrane?
Oof, okay. That is a big question.
You
yes.
can thank me later for that question.
So absolutely, the tight junctions have a very unique lipid composition. They're lipid raft-like structures, and that lotus root morphology that you see actually is mirrored by differences in lipid composition, but also lipid curvature. So there's a wonderful paper from Genalia Farm in Science from earlier in the year, I think, which looked at tight junctions, and you can see that there's marked differences in the curvature across the tight junction. So that lotus root looking appearance is mirrored by lipid composition, but also lipid curvature. So it's possible that maybe lipid curvature is part of it as well as lipid composition.
Very good. Question from Jeff Leach. Is TNF established as the main or only trigger?
No, not at all. So a variety of inflammatory stimuli can stimulate necroptosis, interferons, viral zRNA, endogenous zDNA. So there's a variety of stimuli. Interestingly enough, in the supplementary to our paper, the clusters that we see with TNF-induced necroptosis are far less apparent with other stimuli. So I don't know whether that's got to do with the strength of stimulation or whether there's fundamental differences in the resultant necrosomes, but that's something that would be really cool to follow up.
Very good. Another question, have you had a look in vivo or ex vivo tissues such as the mouse gut or the human gut, I could add?
So the only thing we've looked at preliminarily is the human organoids, and the data is not conclusive as to whether we've got decent immunofluorescence staining for MLKL, RIP3 and RIP1 in those. The reagents that we use for mouse necroptosis have only recently become available, and we hope to, like I said, put the paper on bioarchives next week. So I haven't looked at mouse necroptosis in tissues until very recently, but yeah.
Yeah, that Antibodies brand spanking new. So yeah, looking forward to doing some cool stuff with it. Chris Tonkin says, fantastic work. Anna Voss asks, what is the ultimate fate of a cell when you inhibit trafficking of MLKL to the plasma membrane? Does it find another way to die?
Yeah, great question. So yeah, where there's a will, there's a way. So I was careful to mention that these are rate limiting checkpoints. They slow down necroptosis, but the fact of the matter is, if you have sufficient flux through that core signaling axis, you get sufficient phosphorylation, oligomerization, and membrane association of MLKL, the cells eventually die. So if you wait 24 hours and you look there, the slowing down those trafficking pathways, those cells are still mainly dead. So only if you directly inhibit MLKL with something like NSA do you get long-lasting protection from necroptosis. So yeah, so these are rate-limiting checkpoints, but they're not panaceas to stop necroptosis.
Thanks for that. I think this might be the last question from Jake, who asks, might have missed this, but have you ever seen MLKL clusters in endosomal trafficking pathways given the Golgi association?
That's a more complicated question than first at a glance. So MLKL has been reported to actually constitutively control endosomal trafficking, and there's also reports of MLKL directly associating with parts of the internalization machinery like the flotillins or other escort machinery. So there's plenty of evidence to suggest that MLKL can and does associate with the endosomal compartment. We have been actively trying to find co-localization of phospho-MLKL during necroptosis with those compartments and struggled. Although because we have to use very particular fixative conditions, it could just be that we're technically not able to pick up the routinely used markers to get that really nice co-localization. So we've tried, haven't got it yet, but I wouldn't be surprised if we can get it in the future.
Yeah, those are the questions. Thank you so much. Oh, hang on. We've got one late runner here from Rory Bowden. What about cells that don't form tight junctions? Do they have the same MLKL effector mechanisms?
So those two, there are, so MOLT4, which is a non-adherent leukemia cell line, I think, doesn't form junctions, doesn't express Z01, but does have those same trafficking and threshold checkpoints that I talked about. So you don't need to form tight junctions for those two pathways to be in operation. That being said, there's definitely a trend for these pathways being more important in adherent cells that do form junctions. So I think maybe 5 of the 7 cells that we assayed use those junctions, and that's in the sup of our paper as well.
Okay, it doesn't stop though. There's more love. Misty says, terrific talk. Someone has asked also, what do you think is passing between cells?
So given how much MLKL, I mean, it's really surprising the amount that MLKL is able to accumulate at a cell before it kicks the bucket. So at least in, for example, the HC29 cells, the threshold for membranolysis seems to be, in my view, very, very high. So to get sufficient amounts of MLKL to go from one dying cell to reach the threshold on a neighbouring cell, I think's it's pretty far-fetched. I don't think you're going to get a lot of MLKL, and same goes for the sort of the idea that MLKL is released in exosomes, and then those exosomes can then dock with other cells and cause them to die. I just don't think the threshold requirements for MLKL are so high that you're not going to get enough to kill the cell. But I can envisage a scenario where MLKL from a neighbouring cell perturbs the membrane and sends a signal, a danger signal maybe to the neighbouring cell. So I think that is at least an interesting idea, but we don't have data to that.
Yeah, I think it's fair to say there's a few gaps in knowledge there, because if you add recombinant MLKL to cells, nothing happens. So there's certainly a contextual thing. But we'll draw a close there. Thank you so much, Andre. I'm sure everyone's applauding at home. But thank you so much.
Thanks, guys. Cheers.